Okay, so let's start. Let's cover the content for the current module, module 11, which is covering chapter 10, blood, and chapter 11, the cardiovascular system. So there was a question, can you get the condensed slides? So what you can do with the slides is you download, what you should do is download Adobe Reader. And from the Adobe Reader, so download the slides, like save them onto your computer. Then you can open them into Adobe Reader, and Adobe Reader allows you to print multiple slides per page. I have an actual video that I did on that, and I'll email that. It's on Blackboard, but I'll email that out to you as well. Email instructions on printing multiple slides per page. It's real easy. You just have to download the slides, and then you open Adobe, and you then open those slides in Adobe, and then you have the options. You can print 4 per page, 6 per page, and ridiculous number per page if you so desire. So let's move to okay. So using your muddiest points, I put together today's presentation on what we're going to cover. However, I'm flexible. Certainly, I understand that everyone who showed up today is not everyone who more people, I should say, more people um, participated in the Muddiest Points Wiki than have shown up today. So what the Muddiest Points Wiki reflects, and what you need, that is the group of students who have joined me on online today, may not coincide completely. So I might ask from time to time, hey, this is this worth us talking about in the limited amount of time that we have? Our first chapter was chapter 10, blood. And all right, this was the first screencast where there was some muddiest points. It was on erythrocytes, specifically explain how erythropoiesis is controlled, and then describe anemia, polycythemia, and the causes and effects on hematocrit. So let's talk about that. So erythropoiesis is the production of red blood cells. Red blood cells are continually being produced by the red bone marrow found in bone. What stimulates the production of red blood cells? Well, it's erythropoietin, that is a hormone that's produced by the kidneys. What causes the kidneys to release erythropoietin? It's a reduction in oxygen levels in the blood. So let's say your oxygen levels drop for whatever reason. That stimulates the kidneys to release erythropoietin. Erythropoietin causes the red bone marrow to produce more red blood cells. As red blood cells levels increase, now you have a greater oxygen carrying capacity of your blood, and so the oxygen levels hopefully return to normal. When that happens, that stimulus that caused the kidneys to release erythropoietin is no longer there, and so there's a reduction in the release of erythropoietin, and there's a reduction in the production of red blood cells. That is how red blood cell production is increased or decreased. That's how it is controlled. Okay. Now, if you understand that, then what's the effect on hematocrit? Well, first, let's talk about what hematocrit is. Hematocrit is the percentage of red blood cells in whole blood. So if one were to take a sample of your blood and they'd have to put it in a heparinized needle, we have to have an anticoagulant, otherwise the blood would coagulate. And so if we withdrew blood from one of your veins and we placed it in a test tube, and then we place that test tube in a centrifuge to spin it down and separate the components by density, we'd find that the red blood cells drop to the bottom. And if we if we calculate the percentage of red blood cells in your whole blood, that would be hematocrit. So as red blood cell production increases and the red blood cell numbers increase, that will increase your hematocrit. Polycythemia is if you have an above normal level of red blood cells. What could cause that? Well, if you had cancer of the bone marrow, where for whatever reason your bone marrow is putting out an abnormally high number of red blood cells, that could cause it. If you live at a high altitude, well, what would an increase in the altitude at which you live have on red blood cell production? Well, as you go higher and higher in altitude, the partial pressure of oxygen drops. If the partial pressure of oxygen drops, then the oxygen level in your blood is going to drop. If the oxygen level in your blood is going to drop, that's going to stimulate the kidneys to release erythropoietin, which is going to cause increased production of red blood cells to raise the oxygen content of your blood back to normal levels. So that, just living in high altitude, can increase hematocrit. And if you have impaired lung function, like if you have a chronic respiratory illness where you can't oxygenate your blood as someone would normally, like if you have chronic bronchitis or emphysema, then that could cause an increase in your hematocrit above normal, polycythemia. Anemia is if you have a below normal level of oxygen carrying capacity in your blood. There are different causes of anemia. One is iron deficiency, what's called iron deficiency anemia. If you're not getting enough iron in your diet, or if you're losing a lot of iron through hemorrhage, then that could cause a decrease in your hematocrit. Iron is a component of hemoglobin. If you don't have enough iron, your bone marrow can't make, well, not your red bone marrow, but the body can't make sufficient amounts of hemoglobin. So you're going to have a decrease in hemoglobin in your red blood cells, and that's what carries oxygen. So you're going to have a lower oxygen carrying capacity of your blood. Vitamin B12 or folate deficiency can also cause anemia. These vitamins are involved in the production of DNA. Why does that matter? Well, your red blood cells die in about 150, 120 days. So you have to constantly be producing new red blood cells. Well, in order to produce new red blood cells, stem cells have to constantly reproduce, meaning mitosis and cytokinesis. Mitosis and cytokinesis require replication of that DNA. That requires vitamin B12 and folate. If either one of those are deficient, then there's going to be a decrease in normal replication of that DNA. That's going to lead to a decrease in the production of red blood cells, which is going to lead to a drop in your hematocrit and a decrease in the amount of oxygen that the blood can carry. And sickle cell anemia is a genetic disorder where you don't produce normal hemoglobin. It causes the cells to sickle and they don't carry as much oxygen. Sickle cell anemia, I don't think would have much of an impact on hematocrit, but the cells don't carry as much oxygen. So before moving on to the next topic, I want to ask, are any questions about erythrocytes, erythropoiesis, anemia, polycythemia, and hematocrit? Okay, so let's move on to the next topic, which is platelets and hemostasis. And let's see here. Students asking this are Scott, Farrell, Katie. And Katie is with us, so we will definitely ask those questions. Okay, so I am not sharing my screen. Please bear with me right quick so I can share my screen again. No, stop it. Stop it, stop it, stop it. I think I'm there. And I apologize, Chrome is not responding at this time. It should respond in just a moment. We're going to move to platelets and hemostasis, I hope. Chrome is stuck. I don't know if you can hear me, but Chrome is stuck right now, and I might actually have to shut down I might have to close Chrome and then come back into it. So I might lose you, and okay, thank you for your patience. That had never happened before, but I guess it was about time. Okay, so next topic were platelets and hemostasis. So let me find the way. And really the only question that was asked had to do with clinical uses of anticoagulants. And so what anticoagulants are, they are substances that block the coagulation of blood. And the only thing I really wanted you to understand was that there are some individuals that are prone to get blood clots. And when I say blood clots, I mean abnormal blood clots, a thrombosis, where there is no damage to the blood vessel. You only really want blood to clot when there's damage to a blood vessel, because the whole purpose of blood clotting is to promote hemostasis, which is the stopping of blood when there is damage to a blood vessel, because you don't want to lose too much blood from your cardiovascular system. However, there are some individuals that are prone to blood clots, and that can cause people who have cardiovascular disease are prone to blood clots, and that, of course, can cause stroke or a cerebral vascular accident. If it occurs in blood vessels of the head, you can get a pulmonary embolism, where a blood clot then breaks off and then lodges in the blood vessels of the lungs, and that can be fatal because it stops blood flow to the lungs, and so there's a drop in oxygenation of the blood, and it can also occur in the coronary circulation, which of course can lead to a myocardial infarction or heart attack. And so there are medications that are prescribed and some that you can take over the counter that are used to reduce the risk of thrombosis. And some of them you're already familiar with, like aspirin, ibuprofen. Those are coagulants. They're not very potent anticoagulants, but so typically people who are, they'll recommend you take an aspirin a day or ibuprofen a day for people who are like over 70, that sort of thing. But then there are others like Aspirin and Dimecol, which are quite potent, and people who are really at risk of blood clots may be prescribed those for a period of time. And that's all I really wanted you to know with respect to the anticoagulants. Now, if you take something like heparin or Dimecol, you have to be very careful, because if you cut yourself or damage a blood vessel, you can have excessive bleeding because it takes longer for the blood to clot. Any questions about that, or any other questions about platelets, hemostasis? So there were a couple of questions about blood typing and blood groups. I don't know why that didn't appear on here. Guess I neglected to indicate that. But Katie and another student had some questions about blood typing, so I thought it'd be appropriate to talk about blood typing. And what I find is blood typing is something that students have an interest in, and they don't really know a whole lot about it. So let's talk about that really quick. And I also am going to share with you a nice online game that you can play, which I think is really cool. So let's talk about blood groups. So there are somewhere in the order of like 20 different blood groups, but the two blood groups that cause the most significant rejections, if an individual is transfused with incompatible blood, are the ABO blood groups and the RH blood groups. So those are the two that your book discusses and that we're going to talk about here. The ABO blood group is based on the presence or the absence of two antigens. If your let me talk about what an antigen is. Let me pull this up. Okay, so if you recall, when we talked about cells back in the Cell and Tissues chapter. We mentioned that there are glycoproteins that are embedded in the phospholipid membrane, the biphospholipid membrane of the plasma membranes of cells, and that some of those glycoproteins act as like "hello, my name is" stickers, so that your cells can recognize your self cells versus foreign cells, right? You want your immune system to attack foreign cells, but you don't want them to attack cells that belong to you. Well, those glycoproteins that do that, those self-recognition glycoproteins, are referred to as antigens. If your immune system recognizes them as being self, then they're called self-antigens. If they don't recognize them, then they're considered foreign. Okay, so you have all these antigens that are on the outer surface of your plasma membrane. As I mentioned, there are over 30 different antigens on a red blood cell. I said 20, but they're actually 30. But as I said, only the ABO blood group and the RH blood group of antigens are the ones that are typically responsible for rejection. So those are the two that we focus on. The ABO blood group is based on the presence or absence of two antigens. If your red blood cells have the A antigen and the A antigen only, you are type A. If they have the B antigen only, you are type B. If they have both the A and the B, you are AB. And if they have neither, you are O. Okay? So I'm A. That's my blood type. So my red blood cells have the A antigen on the surface of their plasma membrane. If you have B, you have the B antigen. If you are AB, you have both the A and the B, and if you have O, you have neither. Now, your immune system produces antibodies. Those are proteins that will attack antigens that your body doesn't have. If you have the A antigen, your immune system will produce anti-B antibodies. Those are antibodies that will attack the B antigen. If you are B, your immune system will produce anti-A antibodies. Those are antibodies that will attack the A antigen. If you are AB, your body doesn't produce antibodies against the A or B antigen. Why? Well, your body would attack itself. Your immune system literally would be destroying its own red blood cells. So if you're AB, you don't have antibodies that will attack the A or the B antigen. If you are O, because you have neither the A nor the B antigen, you have antibodies against both. Now, I'm going to stop sharing for a moment and come back to me because I had an instructor show me a nice little easy way of remembering that. So let's say that this little bottle here represents a red blood cell. And I'm going to take two little post-it notes. See if you can see those. A and B, all right? A and B, okay? And so if you are A, if you're A, your red blood cells have the A antigen, but there's B antibodies floating around, or anti-B antibodies floating around. If, on the other hand, you are B, your red blood cells have the B antigen, and there are A antibodies, anti-A antibodies floating around. If you are AB, your red blood cells have both the A and the B antigens, and there are no anti-B or anti-A antibodies floating around. If you are O, your red blood cells have neither one, and then you have both anti-A and anti-B antibodies floating around. So this is a nice little demo that you can do for yourself to help you remember, hey, the certain blood types, what antigens do they have, where are they found, and what antibodies do they have? Does the body produce? So why do you care about whether there are what type of antibodies are present in an individual? Well, the antibodies determine what type of blood that recipient can receive. If you have anti-B antibodies, you can't receive B blood because those anti-B antibodies will attack it. If you have anti-A antibodies, you can't receive A blood because your antibodies will attack it. If you are O, you can't receive A or B because you have antibodies against both. So you can't receive blood for which you have antibodies against. So someone who's A can't receive B, someone who is A can't receive AB, someone who is B can't receive AB, someone who is O can't receive A or B, and someone who is AB can receive everything because they don't have antibodies against A, B, or AB. So let's talk now about how we type blood, right? So you understand that someone who is A can't receive B or AB bloods. Someone who is B can't receive A or AB blood. So that means that we have to be able to type donor blood before we can give it to the recipient. Well, we have to type the recipient to know what kind of blood they have, and we also have to type the donor to figure out what kind of blood they have. Okay? So let's talk about how we do that. So antibodies are very specific to the antigens that they will attach to and attack. So anti-A antibodies will only attack A antigen. Anti-B antibodies will only attack B antigen. So in a lab, we can use antibodies to probe for antigens. We can, if a blood sample comes into the lab, for me to figure out if the person's A, AB, B, or O, all I have to do is take their blood samples and then mix it with these different antibodies. So let's say I pulled someone in the class's blood. You didn't know your blood type, right? And I pulled your blood and I wanted to determine, hey, does your blood have A antigens? All I'd have to do is mix it with some anti-A serum. That's just some serum that has the antibodies in it. And if your blood cells have the A antigen, it'll clump up because those antibodies will bind to the antigens on several cells and they'll agglutinate or clump up. And I can say with certainty, if I get agglutination, that you have the A antigen. The next step would be, so what that would mean is you are either A or you are AB. So then I would do the same thing again and mix your blood with anti-B antibodies. And if I get no agglutination, then you're A. If I do get agglutination or clumping, then I know you're AB. So let's look at these different patients whose blood samples were used to determine their blood type. So to determine someone's ABO blood group, all I have to do is have 2 little blood samples from an individual, and then I mix one of the blood samples with anti-A serum, and I mix other blood samples with anti-B serum. If I get clumping, then I know that the antigen is there. If I don't get clumping, then the antigen is not there. Okay, so let's say I take I take a blood sample from a patient and I mix it with anti-A. Voila, I get clumping. That means that there is A antigen there. Then I mix it with mix that blood sample with anti-B antibodies, and I get clumping. So the A antigen is there and the B antigen is there. So what's their blood type? If you said AB, you are correct. Another patient, I take their blood, I mix it with anti-A serum, I get no clumping. That means that those antibodies did not attach to an antigen. I then mix a blood sample with anti-B antibodies and I get clumping. What's this person's blood type? You said B, you are correct. Third patient, mix it with the anti-A, get clumping, mix it with the anti-B, no clumping. So this person is A. Final patient, if I take the two blood samples and I mix them with anti-A and anti-B and I get no clumping at all, then that person's O. What you are doing when you mix their blood with the anti-A serum and the anti-B serum, you're just asking, hey, is there A antigen there? Yes or no? Is there B antigen there? Yes or no? And the answer to those questions determines their ABO blood group. Yes, yes, they're AB. No, yes, they're B. Yes, no, they're A. No, no, they're O. What about the RH blood group? And I sort of flew over that, all right? Because everyone knows, I would assume that typically when your blood, when someone speaks of your blood type, then there's always like a positive or negative that follows that. Well, what does that mean? So that's the second blood group that is responsible for the most vigorous rejections, what's called the RH blood group. And it's basically based on the presence or absence of a protein. That protein is called agglutinogen D, but I guess people don't like saying agglutinogen D, so they just call it the RH factor or the RH antigen. Why is it called the RH antigen? Well, because it was first identified in rhesus monkeys, and rhesus begins with RH. There's no other explanation other than that. If some individuals have the agglutinogen D, others do not. If you have the agglutinogen D, you are said to be RH positive. If you don't, you are RH negative. I am A positive. I have the A antigen and I also have the agglutinogen D. When typing blood for the RH blood group, we use the same process. We'll have anti-RH antibodies. We'll have anti-RH antibodies that will mix with the serum. I thought I was sharing my screen until I looked at my other computer. How long have I not been sharing my screen? Was I talking this whole time and I wasn't sharing my screen? Oh, so why didn't anybody say anything? So did you see this screen while I was going through the blood typing? No. Okay. All right. All right. Oh gosh. And nobody said anything. Just sat down and let me talk. All right. So my other class, they've called me out on it when I haven't been sharing. They'd be like, are you supposed to be sharing? I'll be like, oh yeah, I am. I'm sorry. Right. You guys just let me go. All right. So so this was the screen I was supposed to be showing you about blood typing. So we have 4 different patients here, and we're taking their blood and we're mixing them with anti-A serum and anti-B serum. Here on the anti-A serum, if you get clumping, that means there's antigen there. If you don't get clumping, then there's no antigen there. So here we mix a sample with anti-A serum and anti-B serum, and both clump. Therefore, they are AB. Here we mix them with anti-A and anti-B. We only get clumping with B, right? So they're B. Here we only get clumping with A, so they're A. Here we get no clumping at all, so they're O, right? In each situation, each patient, we're asking, is there A antigen there, yes or no? Is there A antigen there, yes or no? If it clumps, means that the antigen, the antibody found antigen. Now let me share with you this really cool this really cool game, blood typing game. And I had it up, but then I had to restart Chrome, and so I lost it. And I have a link to it from Blackboard, but you can also get to it by going Nobel blood typing game. Okay, play the blood typing game. And it'll literally illustrate the basic principles that I just talked about, as you'll see in just a second. This is by far the coolest educational game I have ever come across. So we have a patient here that needs blood. First thing we have to do is type their blood. So we pull some blood, and we're going to mix his blood, and we're going to mix it with anti-A serum, anti-B serum, and anti-RH serum, and look for the clumping. Anti-A serum, we have no clumping, right? The anti-A antibodies do not find any A antigen. Why? Because he's got B antigen, so no clumping. We mix the blood with with anti-B antibodies, and lo and behold, they find B antigen, and so we get clumping. So he has B antigen. Then we do the same with anti-RH antibodies. The RH antibodies finds that agglutinogen D, and we get clumping. So no A antigen, but B antigen and RH antigen. So this person is B positive. Okay. So now that we've correctly typed the blood, now we got to figure out what kind of blood can we give him. Well, if he's B positive, obviously we can give him B positive blood. If he's B positive, can we give him B negative blood? What are you thinking? Let's try it. Yes, we can. Why can we do that? Well, if he has the agglutinogen D and B negative blood doesn't have the agglutinogen D, he doesn't have any antibodies against the RH, so that's not a problem. What else can we give him? Can we give him A? No, we can't, because he's B, so he has antibodies against the A. So we can't give him AB because there's A there. We can't give him AB positive or AB negative. So the only thing we have left are O's. Can we give him O positive? Yes, right? He's positive, so he can get positive. And of course, we can give him O negative. Yes! All right, so real cool game, and it's based on the principles that we just talked about with respect to blood typing. All right. That is correct. All you're doing when you're blood typing for the Rh factor, you're just determining, do they have it, do they not have it? Do they have the agglutinogen D, or do they not? If they do, they are positive. If they don't, they are negative. Again, I am A positive. So my red blood cells have the agglutinogen D antigen. There are no antibodies against it in my body, so I can get A positive from other people donated to me. I can get O positive donated to me, but I can also get negative, because negative just means they don't have the protein, and I have the protein, they don't have the protein. Right? Then I'm not producing antibodies that would reject O blood, because O blood doesn't have B antigens, and O blood doesn't have the agglutinogen D. But even if it's positive, it doesn't matter, because I don't have antibodies against it. Okay, that is all I had for blood. So I'm ready to move on to the cardiovascular system, which is where students tend to have more questions anyway. So unless there's any issues with that, that's where I would like to go next. Okay, so the first muddiest points I had was on the cardiac cycle. Actually, that's not where I had my first questions. Excuse me. Let me stop sharing. That's not where we're going to go. We're going to go to cardiovascular system, which is not appearing. Okay, now it's appearing. Thank you so much. Okay, now it's appearing. First question was on the cardiovascular system, and the first questions were on the function and organization, specifically the flow of blood through the pulmonary and systemic circulations. Okay, all right, so let's talk about that. So your heart is a pump, right? Specifically, the ventricles are what actually do the pumping. The ventricles do the pumping. The atria just receive blood from different parts of the body. But your heart is really two pumps. The right ventricle pumps blood through what's called a pulmonary circuit, basically to the lungs, and then it seeps back. And then you have the left ventricle that pumps blood through the systemic circuit. So as you probably recognize, the color coding of blue reflects deoxygenated blood. That's blood that's low in oxygen, high in carbon dioxide. And the red color coded blood indicates it's oxygenated blood that's high in oxygen, low in carbon dioxide. The right side of your heart is receiving deoxygenated blood from the body, from the systemic circulation. So it pumps deoxygenated blood through what's called the pulmonary circuit to the lungs. Well, why is it going to the lungs? Well, so carbon dioxide can be removed and oxygen can be loaded into the blood. Then oxygenated blood returns via the pulmonary veins to the left side of the heart. So this is the pulmonary circuit to and from the lungs. It goes to the lungs as deoxygenated blood. The blood returns oxygenated. Now that we've oxygenated that blood, we're ready to send it to all the tissues of the body. So the oxygenated blood then leaves the heart via the aorta and enters the systemic circulation, where it's circulated to all the tissues of the body outside of the lungs. At what's called the systemic capillaries, oxygen is unloaded to the tissues and carbon dioxide is loaded from the tissues, and then deoxygenated blood is returned to the right side of the heart via the veins. So that's how the heart is two pumps, and that's how blood flows through the pulmonary circuit and through the systemic circuit. Okay, let's go on and move on to cardiac muscle cells. And cardiac muscle cells are quite a few students who indicated this was the muddiest point. And one of the things I wanted you to do in learning about cardiac muscle cells was to compare the skeletal muscle cells to cardiac muscle cells, because we've covered skeletal muscle cells in the muscular system. So you should be somewhat familiar with them. So I want you to compare the two with respect to, well, how do they generate their action potentials? Or how are action potentials generated in skeletal muscle cells versus cardiac muscle cells? What's the source of calcium muscle contraction in cardiac muscle cells? What's the length of contraction of cardiac muscle cells versus skeletal muscle cells? Right? What stimulates muscle cells to contract, skeletal versus cardiac? And then what's their sources of ATP, right? Are all these the same? Are they different? Right? So turn with me in the handout that I emailed to you to section B, where it says cardiac muscle cells. It looks like this. I emailed you this learning activity last week. Hopefully you completed it. Please turn to section B. That is, it's at the bottom of page 1, but the questions that we're going to cover actually are here on page 2. All right, so maybe I can get a little interaction here. So I'm going to ask some questions, and I'd like for someone to speak up, right? Raise your hand, let me recognize you, and then unmute your mic and give an answer. In skeletal muscle cells, almost all the calcium of intracellular fluid comes from which organelle? Don't everybody talk at once. In skeletal muscle cells, which organelle releases the calcium? All right, go.
The endoplasmic reticulum.
Correct. The sarcoplasmic reticulum, which is a smooth endoplasmic reticulum. That is correct. Now, let's contrast that with cardiac muscle cells. Most calcium comes from the sarcoplasmic reticulum, but there's about 10 to 20% that comes from somewhere else. Anybody know? Anyone? Going once? Anyone? Going twice? The extracellular fluid. There are calcium channels in cardiac muscle cells, or in the plasma membranes of cardiac muscle cells, that also allows calcium to come in from the extracellular fluid. So blood levels of calcium can affect your heart. All right, they can affect how those cardiac muscle cells contract. All right, number 3, which cells contract for a longer period of time? Skeletal muscle cells or cardiac muscle cells, and why? Anyone want to tackle that one? Well, cardiac muscle, but I'm going to move on because I don't want us to run out of time. Cardiac muscle cells contract for a longer period of time because they have to contract long enough for the ventricles to empty. What stimulates skeletal muscle cells to contract? Well, that was motor neurons, right? Motor neurons stimulate skeletal muscle cells to contract, always and forever. Cardiac cells, however, they can be stimulated to contract by several sources. One, of course, motor neurons can cause them to contract, but also they can be stimulated to contract by action potentials they receive from other cardiac muscle cells. Those intercalated discs allow sodium ions to move from one cell to the next. So one cardiac muscle cell can actually stimulate another cardiac muscle cell to contract. And third, some cardiac muscle cells have what's called pacemaker activity, where they can spontaneously open their sodium channels and contract. That is very different from skeletal muscle cells, where they have to be stimulated by a motor neuron. Oops, sorry. Which type of cell has the greatest ability to obtain ATP from anaerobic fermentation, cardiac or skeletal muscle? Well, obviously skeletal muscle cells. Skeletal muscle cells, right? They can produce more ATP anaerobically than cardiac muscle cells. Cardiac muscle cells are dependent on oxygen at all times. That is why a restriction in blood flow through the coronary circulation to the heart can lead to heart attack and death. Okay. All right, so just want to move to cardiac conduction system. So there are quite a few questions about the cardiac conduction system. So let's recognize a couple of things. So the atria contract before the ventricles, right? They contract because they have to squeeze blood into the ventricles. And then the ventricles contract to then pump blood through the systemic and the pulmonary circulation. The heart is composed of hundreds of thousands, if not millions, of cells. In order for the heart to beat and to effectively work as a pump, those cells have to be coordinated to contract in a certain sequence. So in other words, the cells in the cardiac muscle cells of the atria have to contract first. Then the cells of the ventricles have to contract second. And the right side of the heart and the left side of the heart have to contract at the same time. Does everybody agree with that? Therefore, there has to be some way to coordinate the appropriate contraction of those hundreds of thousands of cardiac muscle cells. Think of going to Camp Randall and going, if you've ever been there, to a football game, and if you've ever done the wave, right? So you've got tens of thousands of people there. In order for the wave to be created, the spectators have to stand up and sit down at the appropriate time, right? There takes a certain amount of coordination there. The same thing occurs with the heart. There has to be coordination among those hundred thousands of cardiac muscle cells. And what does that is the cardiac conduction system. It's responsible for coordinating the contraction of those many cardiac muscle cells, right? The cardiac conduction system consists of the following. The pacemaker of the heart is the SA node, or the sinoatrial node. Then there's the atrioventricular node, the atrioventricular bundle, the bundle branches, and then the Purkinje fibers. Right? This is an illustration of those in your book, or from your book, I should say. So the sinoatrial node is here. The sinoatrial node is the natural pacemaker of the heart. So action potentials start here, and they spread through the atria, causing the atria to contract. As the action potential spreads to the atrioventricular node, which is right here, sort of between the atria and the ventricles, it pauses, right? There's resistance to further conduction. Why is that important? Well, we need the atria to have time to empty of their blood. So there is a brief pause, and when I say brief pause, we're talking milliseconds here. And then the action potential is conducted to the AV bundle branches here, down into the bundle branches at the interventricular septum, and then finally to the Purkinje fibers. And so notice that the Purkinje fibers run from apex back toward the base. And that's important because we're going to contraction here first and then moving this way, because these ventricles are driving blood through the vesicles, which are here and back here, right? So we want contraction to go from apex toward the base. Okay, so the intrinsic conduction system ensures that the right parts of the heart contract at the appropriate time. First the atria, then the ventricles, and the ventricles contract from apex to base, because the pulmonary trunk back here, which you can't see, and the aorta are where the blood leaves the ventricles and leaves the heart from the ventricles. We can monitor and chart the movement of electrical activity through the heart using an electrocardiogram. That's where the leads are placed at different parts of the body, and we can actually record the depolarizations and repolarizations that occur. And what you're responsible for knowing are the different components of an EKG. You have the P wave, the QRS complex, as it's called, and the T wave. The P wave is depolarization of the atria. The QRS complex is depolarization of the ventricles, and the T complex is repolarization of the ventricles. So what's missing? Shouldn't there be 4 waves? We have depolarization of the atria, depolarization of the ventricles, repolarization of the ventricles. Well, what's missing is the repolarization of the atria. Well, that's obscured by this QRS complex, so we won't see that on the EKG. So let's look at that EKG in action. So this is the heart slowed down to 3 beats per minute. And then you're seeing the EKG down here, and you're seeing actual movement of the electrical activity through the heart. So follow the electrical activity as I chart the EKG. So first we have our P wave as depolarization of the atria. Here's P. Q, R, S is depolarization of the ventricles. And then T is repolarization of the ventricles. Let's speed this up. Oh, sorry, let's speed this up to 18 beats per minute. P, Q, R, S, T. P, Q, R, S, T. P, Q, R, S, T. You can learn a lot about a patient's heart by looking at their EKG. Okay, so additional questions about the cardiac conduction system. So the next questions were on the cardiac cycle. Right? So the cardiac cycle is basically all of the different, all of the actions that occur or events that occur in a complete beating of the heart. Okay, so let's look at that. So there are 3 stages to the cardiac cycle. You have atrial systole, and by the way, systole is contraction. Diastole is relaxation. So the atria contract while the ventricles are not contracting, they're in diastole. Then after the ventricles are filled, the ventricles contract as the atria begin to relax. And then after contraction of the ventricles, the entire heart is in diastole or relaxing. So let's look at this in action. Okay, so as I said, systole means contraction, diastole is relaxation. First, we're going to have the atria contract while the ventricles are not. Then the ventricles contract while the atria relax, right? And then we're going to have both diastole and systole. All right, so here we have, let's look at number one. So here we have blood in the atria, and blood is flowing from the atria down into the ventricles, right? As blood flows into the ventricles and the ventricles fill, to move that last bit of blood from the atria into the ventricles, the atria contract, right? So we have atrial systole to move that blood from the atria into the ventricles. After that blood fills the ventricles, now the ventricles are going to contract. As the ventricles contract, the atria are beginning to relax. Also notice that the tricuspid and the mitral valves close so we don't get backflow of blood back into the atria. After the ventricles contract, relax, and now we have blood flowing from the atria back into the ventricles. Notice that while the ventricles are relaxed, the semilunar valves, those are the valves at the openings to the aorta and the pulmonary trunk, are closed, so we don't get backflow back down into the ventricles, right? And then eventually we go back to atrial systole. So this is a complete cardiac cycle. Okay, so let's define a couple of terms: preload and end diastolic volume. So at the so preload is the amount of blood that is in the ventricles right before they contract. In other words, it's the volume of blood in the ventricles at the end of the relaxation phase, at the end of diastole. So preload and end diastolic volume are the same, right? Stroke volume is the amount of blood that is ejected when these ventricles contract. So when they contract, whatever amount that is expelled, that is the stroke volume. So what about after these ventricles contract? What's left in the ventricle is the end systolic volume. The ejection fraction is the percentage of blood that was delivered to the ventricle that was ejected. So in other words, if there were, and I'm just pulling these numbers, if 100 milliliters was the end diastolic volume, and after contraction, the end systolic volume was 20, then the ejection fraction would be 80%, because 80 of that original 100 would have been expelled. Okay, right? And here's just another example. If the stroke volume is 70 and the end systolic volume is 50, then the ejection fraction is 58%. All right, let's take a look at your the learning activity, and let's calculate this. So prior to ventricular systole, we had 100 milliliters of blood in the ventricle. Okay, so before contraction, there was 110 milliliters of blood in the ventricle. After ventricular systole, there are 44 milliliters left. So two questions. What is the preload? And what is the end diastolic volume. Okay? I'll give you a second to come up with those answers, and then we'll go over them. Okay. Anyone know what the preload is? Anyone want to take a guess at that one? Well, the preload is 110 milliliters. What's the end diastolic volume? Well, what's the amount after at the end of diastole or relaxation? Well, it's also 110 milliliters, right? This was sort of a trick question just to see if you understood the terminology, right? So prior to ventricular systole, there's 110 milliliters in the ventricle. So ventricular systole means contraction of the ventricles. Contraction or systole occurs at the end of diastole. So the preload, which is the amount of blood in the ventricles prior to contraction, is 110 milliliters because by definition, it's a prior to ventricular systole, there are 110 milliliters in the ventricle. So that is by definition preload, right? And if it is prior to systole, right before systole, it means that it's at the end of the relaxation phase, which is diastole. So the end diastolic volume is also 110 milliliters. Okay, it's the amount of blood that's in the ventricles right before it contracted. That is preload, that is end diastolic volume. Okay, there were a couple of questions about the structure of blood vessels. I do realize I'm gone over, and if you have to leave, you are welcome to do that. Otherwise, I'm going to keep going until we've covered everything, all the muddiest points, and answered any questions that students have. So there are quite a few questions about the structure, or quite a few muddiest points on the structure of and function of blood vessels. Okay, so I'm going to talk about that, and I'm going to talk about, then there was a question about explain how the cardiovascular system directs blood to specific organs and tissues. So I'm going to address both of those. Okay, so this figure here is from your book, and what it basically shows you is the arteries here, artery on the left, a vein here on the right, and then capillaries here in the middle. I want to focus on the artery and the vein, all right? So the anatomy of the artery and vein are going to reflect its physiology, its function. So the wall of an artery and a vein are composed of three tunics: the tunica intima, media, and externa. Let's look at the functions of these three different blood vessels, and it will help you understand the anatomy we're going to talk about in just a second. So arteries are closest to the ventricles. Arteries take blood away from the heart. Arteries take blood away from the heart. So they're closest to the ventricles. So they're going to be receiving blood at the highest pressure. And also, they're going to need to be able to stretch and recoil as the ventricles contract and relax, or doing systole and diastole, right? So they need to be able to withstand high pressure, and they also need to be able to stretch and recoil. Veins, on the other hand, they're farthest away from the ventricles. Blood pressure is low at all times, and they don't have to stretch and recoil like arteries do. And then capillaries are the only location in the blood vessels where there's actual exchange of substances between the blood and the tissues. So we have blood vessels with three very different functions, and that's gonna be reflected in their physiology, in their anatomy. So as I said, these blood vessels, or at least arteries and veins, consist of three layers or tunics. Okay, capillaries actually only have one layer, but we're going to focus on arteries and veins for now. The innermost layer is the tunica intima. The tunica intima basically is the layer that's in contact with the blood of the lumen, right? It's what lines the lumen. You have one flat layer of epithelial, of squamous epithelial tissue. It's very, very, they're very, very flat. They have very low friction, because you want as little friction between the blood and the artery as possible. And then you have some connective tissue that supports the endothelium layer. And then, at least with the artery, you have this elastic tissue that helps the artery stretch and recoil. That is, it return to its normal length. Let's move to the second coat, which is the tunica media. The tunica media is mainly composed of smooth muscle. And notice that in the artery, it's much thicker than in the vein. Do you see that? Much thicker in the artery composed to the vein. And that contributes to the overall greater thickness in the wall of an artery compared to a vein. Notice that although the lumen of this artery is much smaller than the lumen of this vein, the wall of the artery is much thicker, and that's mainly due to the increased thickness of the tunica media, specifically the smooth muscle in the wall of the tunica media, in the tunica media of the artery. Why does an artery need to have more smooth muscle compared to a vein? Or what ability does that give the artery? Well, arteries, because they have a thicker layer of smooth muscle, that allows them to vasoconstrict and vasodilate to a much greater extent compared to a vein. So to understand how smooth muscles allow for vasoconstriction and dilation, I want you to imagine that you and other classmates are standing in a circle holding hands, right? And we're going to be the smooth muscle cells, right? We're holding hands. Let's say we're now going to, we're standing in a circle holding hands, right? We're representing, as smooth muscle cells, the wall of the blood vessel, the artery. And the middle of the circle would be like the lumen. If we contract means to shorten, we pull our hands in to touch our hips. Well, if everyone in a circle does that, that's going to cause the circle to get smaller. We're all going to have to take a step forward, right? And that's going to cause the circle to get smaller. That is vasoconstriction. That's what happens when your blood vessels constrict. If we then decide we're going to keep one another, we're going to hold hands, but we're going to extend our arms, and we take a couple of steps back, that's going to cause the circle to get larger, right? That's what happens with vasodilation. Because of the greater, thicker layer of smooth muscle in arteries, they have a much greater ability to vasoconstrict and vasodilate. Lastly, we have the, and I should say the tunica media of the artery also has another layer of elastic tissue. And lastly, on the outer portion of the blood vessel, we have the tunica externa, and basically its function is to protect the deeper tissues. And they look pretty much the same in both the arteries and the veins. Okay, lastly, the capillaries. So capillaries only have one tunic, and that's basically the tunica intima. They basically consist of simple squamous epithelial tissue and the basement membrane. Recall, capillaries, it's substances have to move across the wall of the capillaries. So, and much of that happens by diffusion. And so you have to have a very thin surface to facilitate diffusion across it. Okay. So now let's address the question about how the body is able to increase blood flow to certain parts of the body and decrease blood flow to other parts of the body. Well, I should say how the cardiovascular system is able to increase blood flow to certain parts, certain organs and tissues while decreasing blood flow to others. So this is a capillary bed here. A capillary bed is fed by small arteries called arterioles, and it's drained by small veins called venules. Okay, so this is our capillary bed, all these capillaries coming off here. Now notice that at the opening to all these small or true capillaries, we have little sphincter muscles. A sphincter muscle is nothing more than a circular muscle, like the muscle around your mouth, okay, or your eye, allowing you to squint and a whistle, right? When these sphincter muscles are relaxed, we get blood flow through these true capillaries. Right? When you're running, this would be the situation with the capillaries in your the muscles of your legs. However, my guess is most of you are sitting down right now. You're not really using the muscles in your legs to do much. So what's happening there is probably those little sphincter muscles in the capillaries of your leg muscles are contracted. And there's decreased blood flow to those capillaries, and instead it moves through what's called the vascular shunt, right? Because you're not walking around, you're not really using those skeletal muscles in your legs much. There's minimal blood flow there. It's just enough to get the oxygen that they need, keep them alive, right? But there's more blood flowing to other parts of the body, like your digestive tract, urinary tracts, right? Brain, that sort of thing. So this is a mechanism by which your body can direct blood to those tissues that are more metabolically active than others at any given time. Okay, I'm going to move to the next topic, which was blood pressure. Right? There were questions about using the equation to explain the effects of cardiac output, peripheral resistance, and arterial blood volume on blood pressure. So let's talk about that. Okay, so what is blood pressure? Let's make sure we understand what that is before we go any further. So blood pressure is basically the amount of force that the blood exerts on the walls of your arteries. That's what blood pressure is. It's the force applied per unit of area by the blood on your arteries. The equation for blood pressure is blood pressure equals cardiac output times peripheral resistance. Cardiac output is the amount of blood that leaves the heart per minute. Peripheral resistance is the resistance to that blood flow. We could illustrate these components of blood pressure by this little figure here. cardiac output. So these are your arteries. We measure blood pressure in the arteries. So this represents the arteries. And these little arrows are pressure that the blood is exerting against the walls of the arteries. Cardiac output is shown here. So blood is flowing into the arteries from the heart. Resistance to blood flow out of the arteries is mainly in the form of these arterioles. That's what these little vice grip is meant to sort of represent, right? This is resistance to blood flow out of the arteries. So if we relate these components to this equation, the greater the cardiac output and the greater the resistance to blood flow out of the arteries, the more pressure you're going to have here, right? That just sort of makes sense, right? The greater this is, the greater the input and the greater the resistance out, the more blood you're going to have here and the greater your blood pressure. So let's look at some examples of like low blood pressure, high blood pressure, right? So in low blood pressure situation here, we have low cardiac output and we have low peripheral resistance. Low peripheral resistance, right? So maybe you're just maxing and relaxing and your blood vessels, your arterioles are not constricted. They're dilated. So we've got little cardiac output, we've got little peripheral resistance, and so we don't have a lot of pressure here. This is low pressure. Now, let's say we increase cardiac output, right? So now the amount of blood flowing into your arteries is greater. And at the same time, we increase peripheral resistance. There's vasoconstriction of these arterioles. So now we have more blood entering the arteries, and there's resistance to blood leaving the arteries. So the volume of blood in the arteries increases, and blood pressure increases. So to understand what affects blood pressure and how it affects blood pressure, you just have to understand what affects cardiac output and what affects peripheral resistance. So what I did here on this, this is another component of the learning activity I gave you, where what I did was I gave you a bunch of different situations and I say, hey, draw an arrow, a up or down arrow, showing the effects on cardiac output, peripheral resistance, and therefore blood pressure. And in many cases, these things may affect more than one, but they affect one more than another. All right, so that's going to be where we focus on what does it affect the most. Let's look at increased heart rate, right? How is increased heart rate going to affect cardiac output? Well, an increase in heart rate, the cardiac output is heart rate times stroke volume. So as heart rate increases, right, it's definitely going to increase cardiac output. Will there be an increase in peripheral resistance? Well, because you've got more blood flowing into the arteries, yeah, but most of the increase is going to be in cardiac output. So there's definitely going to be an increase in blood pressure if your heart rate increases. Well, what about hemorrhage? What if you are losing blood, right? You got cut and you're losing blood from your cardiovascular system. Well, if you're losing blood from your cardiovascular system, that's going to affect cardiac output. It's probably not going to have, it may decrease peripheral resistance a little bit, but it's certainly going to decrease cardiac output. So there's going to be a drop in blood pressure. In fact, if you lose too much blood, the problem is your blood pressure drops so low that your cardiovascular system can't provide sufficient circulation to your vital tissues and you go into shock and die. What about dehydration? You lose more water than you take in. Well, that's going to drop blood volume. So again, less cardiac output, less peripheral resistance. That's going to lead to a decrease in blood pressure. What if we increase blood volume? Well, that's the opposite of dehydration, right? Increasing blood volume, that's going to increase cardiac output. There's going to be a small increase in peripheral resistance. That's going to increase blood pressure. What if we do block a major vein? How would that affect cardiac output? Well, if we block a vein, there's going to be less blood returning to the heart. That's going to lead to a drop in cardiac output, which will lead to a decrease in peripheral resistance, although the biggest effect is cardiac output. That's going to drop blood pressure. Substance that open calcium channels. How's that going to affect the heart? Well, remember that 10 to 20% of calcium in a cardiac muscle cell comes from the extracellular fluid. So blocking calcium channels is going to decrease the calcium content of the cytoplasm. So it's going to decrease the number of myosin heads that interact with the filaments. So the heart's not going to beat as hard. So that's going to decrease cardiac output, decrease resistance a little bit. The biggest effect is going to be the cardiac output and decrease blood pressure. I just said that backwards. I am sorry. If we open calcium channels, it's going to be more calcium entering the cardiac muscle cell. That's going to cause greater contraction, greater blood pressure. If we were to close calcium channels, we were to administer a substance to a patient that closes the calcium channels, then we would get less calcium in the cytoplasm and reduce cardiac output and blood pressure. What about vasodilation of arteries? We make those arteries larger, right? That's going to decrease peripheral resistance and decrease blood pressure. If we vasoconstrict those arteries, that's going to cause an increase in peripheral resistance and an increase in blood pressure. What about scar tissue on the inner linings of arteries? What if there's a buildup of, plaque or calcium deposits on the linings of your arteries? Well, that's going to resist blood flow, right? So that's going to increase peripheral resistance and increase blood pressure. All right. So before we move on, I want to ask if there are any questions up to this point. I feel like I've just gone on and on and on, and there's been very little interaction, very few questions. No one has any questions at all? So everything's making perfect sense. Okay, so the last topic I want to cover is capillary exchange. Oh, got a question. So the question is about diastolic versus systolic pressure. So whenever pressure is given, systolic is always given first. And I know I had a slide on that somewhere. Bear with me just a second. Maybe I can find it. Maybe I can't. But systolic is up. Here we go. I think. So systolic pressure is the peak pressure developed in the aorta and arteries during ventricular systole. So in other words, when the ventricles are contracted. Diastolic pressure is the pressure basically when the ventricles are relaxing, when they are in diastole, right? And yes, and I know I have it somewhere, but yeah, the systolic is always given first. So the first number is always greater than the second number. Oh, here it is. Oh, okay, I'm not sharing here. I thought I was sharing, I wasn't sharing. Now I'm sharing. Okay, yes. The it's always reported systolic over diastolic. Sorry it took me so long to bring that up. Okay, final topic is capillary exchange. And basically with capillary exchange, we're looking at how do substances cross between the capillaries and the tissues of the body. So in this example here, or this figure here, what you're looking at is here are tissues, here's our capillary containing our red blood cells. Our capillaries are bringing in oxygen and nutrients and carrying away waste products and carbon dioxide and substances like hormones that the cell is producing. Substances have to move across the walls of the capillaries to get to and from the cells. What capillary exchange is describing, well, how does that occur? How do certain substances cross the wall of the capillary? So there are 4 routes by which substances cross the walls of the capillaries. First, some can just diffuse across the wall of the capillary. So the capillaries, the wall of the capillaries are composed of simple squamous epithelial cells. Each of those cells contains a plasma membrane, of course, and anything that can diffuse across the plasma membrane of those cells can diffuse across the wall of the capillary. So what do we know that can diffuse across the plasma membrane? Well, anything that's nonpolar, respiratory gases like carbon dioxide and oxygen, lipids, they can diffuse right across. Okay. Also, some substances can also cross the cross the walls of the capillaries by endocytosis and exocytosis. Endocytosis on one side, exocytosis on the other, right? So let's illustrate that. So this is sorry. Okay, so this is a capillary, right? So here's the lumen of the capillary. So here's where the blood would be. This is the interstitial fluid. So down here in the corner would be like your cells, right? So how do substances get across the wall of the capillaries, right? Well, some substances can just diffuse right across the cell. So these are your endothelial cells here, right? Shown here in purple. Lipids and respiratory gases like oxygen and carbon dioxide, they can just diffuse right across. Other substances can cross by endocytosis on one side and then exocytosis on the other, right? Endocytosis on one side, exocytosis on the other. Also, notice that there are spaces between endothelial cells, and these are called intercellular clefts, and substances can move between the cells. So in most capillaries of the body, substances can also move between the cells through these intercellular clefts. Now, not all capillaries, I'd say most, but not all capillaries have intercellular clefts. capillaries that make up the blood-brain barrier would not have intracellular clefts. Those cells have tight junctions between one another. So that would not happen in all capillaries, but most capillaries would. Some capillaries are very, very porous, shall we say. They actually have these large spaces called fenestrations or pores between their cells that allow substances to move between them. You find these fenestrations in places where there's a lot of absorption into the bloodstream, such as the small intestine, where nutrients are absorbed, or around endocrine organs like your pancreas or your thyroid. So again, you wouldn't find these fenestrations in the blood-brain barrier, the capillaries of the central nervous system. Okay, let me go back for just a second. So now that you know the different routes by which substances can cross between the lumen of a capillary and the interstitial fluid of tissues, let's talk about, well, what's going to drive fluid one way or another? What's going to drive the fluid of the capillary toward the interstitial fluid, or the fluid in the interstitial fluid toward the plasma that's contained in the capillary? And that's and that's what's called flow. So we've got this image is not in your book. It was in a previous edition. It's not in your current book. And basically it's showing here we have a capillary, and outside of it is interstitial fluid. So blood is flowing from left to right. So this is the arterial side of the capillary. This is the venular side of the capillary. And then these arrows show pressure, either pressure pushing fluid into the capillary or pressure pushing fluid out of the capillary. Osmotic pressure is pushing fluid into the capillary. Blood pressure, shown here in blue, is pushing fluid out of the capillary. Okay, and we covered what blood pressure is. Let's remind ourselves what osmotic pressure is. So osmotic pressure is the pressure of osmosis, right? Basically, substances that are charged attract water. If we were to put, for example, this container of pure water, well, I should say, excuse me, this container with saline solution, that's water and salt, and we put it in this drum of pure water, the water is going to move from low concentration to high concentration. So over time, the water is going to move from the drum into the container, and it's actually going to cause the column of water to rise. That's osmotic pressure. It's pushing against air pressure. That's what's illustrated down here. Here we have pure water here. Here we have water and saline. So water is going to move this way. Well, what's going to cause water to move into your blood vessels? Well, you have in your plasma those plasma proteins called albumin. Albumin's always in your blood vessels. They're too big to cross the capillary walls, so they're always going to pull fluid into the blood. Blood pressure, of course, is blood pushing against the walls of your blood vessels. In a similar manner to like a garden hose, there's water pushing against the walls of the garden hose. If you cut a garden hose, the water comes spurting out because there's greater pressure inside than outside. So you've got these two forces opposing one another, blood pushing fluid out, osmotic pressure pulling fluid in. So let's look at those two pressures as we go from the arterial side to the venular side of a capillary. On the arterial side, we have high blood pressure, shown here, 35 millimeters Hg, as we move from the arterial side to, I'm sorry, let me stop before I do that. Focus on the arterial side. I'm sorry. On the arterial side, we have 35 millimeters of mercury blood pressure pushing out. Osmotic pressure, we've got 25 millimeters of Hg, 25 millimeters Hg pressure pulling in. Which one's greater? Well, there's a greater force pushing out than there is pulling in, so there is net pressure out. So in other words, there's a net movement of fluid out of the capillary on the arterial side. Now, let's look at what happens to blood pressure as we move from the arterial side to the venual side. It decreases such that it's decreased to 17 millimeters of mercury on the venual side. Osmotic pressure stays the same. So now osmotic pressure is greater on the venual side compared to blood pressure, which is lower on the venual side. So now there is net pressure in. So there's net movement of fluid in on the venual side. So there's net movement of fluid out on the arterial side and net movement of fluid in on the venual side. Okay, as I said, this figure is not in your book, but I like it for explaining this process. This figure is in your book and it shows you the same thing. This line here, I'm at the bottom of the figure. This line here is osmotic pressure. It is the same from arterial side to venual side. Blood pressure on the arterial side is higher than osmotic pressure, and it drops as you go from the arterial side to venual side. On the arterial side, blood pressure is greater than osmotic pressure, so there is net movement of fluid out of the cell. That process is also referred to as filtration. On the venual side, osmotic pressure is greater than blood pressure, so there is net movement of fluid into the cell, into the capillary. Okay, and that's called absorption. So there is filtration on the arterial side, there's absorption on the venual side. And all you have to know to be able to explain that is to be able to reproduce this graph here. Okay, so that is, we have covered all the topics that were listed as your muddiest points. At this point, I would like to open the floor up for any questions you may have. And they can deal specifically with material, or they can deal with any other, they could be any other questions you may have. And feel free to, I'd love to hear your voices. So feel free to unmute your mics.